Hello everyone, I'm Sneha, third year MBBS student at AIIMS Nagpur, and today we'll be doing previous year questions asked in NEET examination for the chapter Plant Growth and Development. So, plant growth and development is included under plant physiology, and one to two questions are being asked from this particular chapter every single year. Okay, so let's start. We have our question number one, and they've asked the typical growth curve in plants is. So we have our NCRT also side by side. So they have asked us for the typical growth curve, and it is given right here that a sigmoid growth curve is usually seen in plants. Okay, so the or the curve which is seen in plants is a sigmoid growth curve which maintains equilibrium with the carrying capacity. It forms asymptote stage in plants. The rate of growth of a plant or a plant part is not always the same during its lifespan. If it would have been same, we would have got a linear growth curve instead if the growth would have been constant. And for linear curve, we have the mathematical equation L at any time t is equal to L naught plus RT, where L naught is length at time zero and R is the growth rate or elongation per unit time. So if we plot the increase in cell number, which is the growth rate, okay, against time, a typical S-shaped curve is obtained. This is called growth curve or sigmoid growth curve. So this is sigmoid curve, and in sigmoid curve, we have three stages. We have stage one, which is the lag phase, stage two, which is the exponential phase, and stage three, which is the stationary phase. And the expression for an exponential growth curve is W at any time t. WT is actually the final size or the size at any particular time t, which is equal to W naught. W naught is the initial size at the beginning of the period, R is the growth rate, and T is the time of growth. Okay. Moving on to the next question, we have the definition of phytohormones. So, what actually are phytohormones? So, phytohormones are also known as plant growth hormones. So, plant growth regulators are also known as phytohormones. And phytohormones are small, simple molecules of diverse chemical composition. They could be indole compounds, indole 3 acetic acid, adenine derivatives. They can be derivatives of carotenoids or they can be gases. Okay. And they are divided into two categories. One is involved in growth promoting activities, one is involved in growth promoting activities. And examples are auxins, gibberellins, and cytokines. And the other group, they play an important role in response to wounds or stresses of biotic and abiotic origin. Okay. And they are involved in growth inhibiting activities. And the examples of this is ethylene. Ethylene can fit in both of these, but it is considered as a growth inhibiting one. And other one is abscisic acid. So abscisic acid is also growth inhibiting. Okay. So phytohormones, they have given us four options, and option D, which is that the phytohormones are plant hormones which are synthesized by plant, and they act as regulators and they influence physiological processes like growth, movement, etc. And the examples are auxins and gibberellins. So the four options that are given are phytohormones are chemical regarding flowering. No, they are chemical regulating secondary growth. They are hormones which are regulating growth from seed to adulthood. And fourth option is regulators, which are synthesized by plants and they influence physiological processes. Moving on to the next one, leaves of many grasses and they are capable of folding and unfolding. So they can undergo change in shape. So in grasses, leaves can fold and unfold. So they can change their shape. Change their shape means they are flexible. So this is due to what? This is grasses, as we know, grass is a monocot plant. So monocot leaves have some specialized cells in the epidermal cells of their leaves, which are called as bulliform cells. So they have bulliform cells, which are the motor cells. They can lose or gain turgidity, due to which their leaves can show coiling and uncoiling. Okay. So the flexibility in the leaves of grass is due to which cells? It is due to bulliform cells, which is our option number C, specialized bulliform cells. Okay. Moving on to question number 4, we have coiling of garden pea tendrils. So garden pea tendrils, suppose this is a tendril. Okay, this is a tendril. And in case we place an object or any support around it, what will it do? It will coil around it. Okay, so it will coil like this. So this shows coiling around the support. And this is due to what mechanism? This is due to our thigmotropism. Okay, so thigmotropism is the mechanism due to which shows coiling of tendrils. Okay, in response to any touch stimulus. Okay, so let us know some more information about thigmotropism. So the coiling of garden pea tendrils around any support is an example of thigmotropism. And thigmotropism is the growth movement in response to touch. So what is the stimulus here? It is touch. And the stem as well as the tendrils of the climbers are positively thigmotropic in their response. So thigmotropism can also be of two types. Thigmotropism can be either positive thigmotropism or it can be negative thigmotropism. So positive thigmotropism is shown by climbers. So suppose this is a pea plant, this is a tendril, and this is support. So the tendril will grow towards the climb and towards the support. Okay. But there is a chui plant. You must have heard about it. So chui plant, when we touch the leaves or the flowers, they tend to shrink away. They tend to contract. So this is an example of negative thigmotropism. So thigmotropic movement, again the same question. So question number 5. Thigmotropic movement is what? It is response to touch stimulus. Response to touch stimulus. And it is the definition for thigmotropism. And it is best shown by what? It is best shown by movement in tendril. Okay. So movement in either tendril or stem of climbers. Right? The same statement we saw in the previous question, but that was a different thing that was being asked. So the phenomena of thingmotropism is a response to contact with solid object. It is exhibited by tendrils of climbing plants that grow around a pole or a stem of another plant. You might have also seen grapevines growing around a support like this. Okay. So clinostat, question number 6, and it was asked in 1993, long ago. So at that time, we used to have a different version of NCRT. There were much more information than we see right now. So they have asked clinostat. So let us know about clinostat. So clinostat is an apparatus which is used to nullify the effect of geotropism. What is geotropism? It is the movement shown with respect to gravity. So it can be either positively geotropic or it can be either negatively geotropic. Roots are actually positively geotropic, that they grow towards gravity. Whereas stem is usually negatively geotropic, that is, they grow away from gravity. Okay, so clinostat is actually an apparatus which is used to nullify the effect of geotropism. It has a clock on the main axis, which is attached to a rod. So the top of rod is attached to a flower pot. The clinostat is kept in horizontal position and allowed to rotate, and the plant growth is observed. Okay. So what is clinostat? It is employed to study about our growth movements, which is irrespective of gravity. So clinostat is used to study plant growth movements in absence of gravity. Okay. Moving on to the next one. What is the site of perception of photoperiod, which is necessary for induction of flowering in plants? Okay, so what is photoperiod? We have, wait just a minute. I'm not moving here. So during flowering, we have question number 7, that during flowering, photoperiodic stimulus is perceived by the leaves of plants. So the leaves are the site of perception of photoperiod. So in 1920, there were two scientists who published their discoveries on photoperiodism and felt it was the length of daylight that was critical. But it was later discovered that the length of the night was the controlling factor. So what do they actually mean? There are some plants that grow in winter. There are some plants that grow in day. So one factor is the duration of day and night. So growth, one factor is the duration of day and duration of night. So, according to the current studies, we have find out that duration of night is more controlling factor. And where is this stimulus perceived? It is perceived at leaves. Okay. So the site of perception of photoperiod is actually the leaves, which is option number D. Moving on to the next one, we have that through their effect on plant growth regulators, what do the temperature and light control in plants? So we have studied about the effect of photoperiod. Now we'll study the effect of temperature and light. So temperature and light, they actually induce what? They induce flowering. Okay. So red light, it promotes flowering. However, it also encourages vertical plant growth. So if we consider this as a plant, so this vertical growth as well as flowering, it is regulated by which light, or which light promotes both of these? Red light. Okay. However, too much red light at the wrong time will cause your plants to stretch. There will be too much vertical growth, which results in lower yield and lanky or unstable plants. Blue light is your secret weapon for amping up the concentration of oils and resins. Okay, so blue light is for what? Increased concentration of oil and resin. Okay, moving on to the next one. Vernalization stimulates flowering in which plant? So vernalization is actually the firstly we'll study about vernalization. We have question number 9. Vernalization is a process in which plants are exposed to lower temperature. Okay, which promotes their growth and flowering. Okay, so plants are exposed to cold on lower temperature. So we have the solution right here that carrot is a biennial plant in which flowers in the second they flower. So carrot grows in 2 seasons. This is the first season and this is the second season. Okay. And when does the flowering takes place? It takes place in the second season. So flowering it occurs in second season. So vernalization is the subjection of seeds or seedlings to low temperature in order to hasten the plant development and flowering. So we expose the seeds. So the seeds are exposed to low temperature. And vernalization, it is commonly used for crop plants such as winter rye and is possible because the seeds and buds of many plants. So rye, it requires the seeds and buds of rye, they require cold in order to break their dormancy. So winter cereals are their food. They are sown in autumn for flowering the following year. However, if the germinating seeds are artificially vernalized, they can be even sown in spring season because we are giving the cold temperature artificially. So biennial plants such as carrot will remain in their long non-flowering rosette until they are subjected to cold treatment. So carrot, even if we grow it in spring, we can give artificial vernalization. Okay. So that its crop yield can be increased. Okay. So moving on to the next one. So photoperiodism was first characterized. Question number 10th. So photoperiodism, we know what is the site for perception of photoperiodism. We just saw it was the leaf. So in which plant did we first characterize photoperiodism? It is a fact-based question that the photoperiodism was first characterized in tobacco plant. Okay. So next question we have that the importance of day length in flowering of plants. So importance of day length is also what? It is the other name given for photoperiodism. So photoperiodism in flowering of plant was first shown in which plant? We just saw that it was tobacco, and it was first studied by which scientists? It was studied by Gardner and Allard in 1920. So the variety of tobacco that they used was Maryland Mammoth, and it could be made to flare in summer by reducing the light hours with artificial darkening. It is originally a tobacco plant is grown in winter, but if we decrease the duration of the time in which it is exposed to light, we can artificially grow it in summer also. Okay. Moving on to the next one, the treatment of seeds at low temperature under moist condition to break its dormancy is called as. Okay. So, question number 12, we have seeds at low temperature and we are giving it moist conditions. So these are the different methods: vernalization, stratification, and scarification. These are the different methods by which we can break the dormancy of a particular seed. Seeds are in dormant phase, so whenever we give them conditions to break that dormancy, then only they'll grow into a plant. So the treatment of seeds at low temperature and under moist conditions to break dormancy is known as stratification. The treatment of seeds at low temperature when there is no moist condition, it is known as vernalization. And stratification, it includes both of these conditions. It includes low temperature as well as moist conditions. Okay. Moving on to the next one. One set of plant was grown at 12 hours day and 12-hour night period cycles, and it had flared, while in the other set, the night phase was interrupted by a flash of light and it did not produce flare. Under which one of the following categories would you place this plant? So we have question number 13. So this is one flower, this is one plant, and this is the other one. We'll take the other one. Okay. So in this, we have 12 hours of 12 hours of light and 12 hours of dark. And in the other one, we have the night phase was interrupted. So light phase is increased and the day night phase is actually reduced. So short day plants require long and uninterrupted. So when they have reduced the night phase, it will what it has actually decreased its production. This has decreased its production. Okay. So this is what? This is a short day plant. So short day plant, they actually require long dark periods. Now, when we decrease the dark period, its production decreases. So it actually needs long dark period. So if long dark period is needed, less light period is needed. And these plants will not flare if the night was interrupted by a flash of light. So these plants are known as short day plants. Okay. Which is option D. Moving on to the next one, we have the pigment that absorbs red and far red light in plants. And this particular question has been asked three times in 1995, 1997, and 2002. So the pigment that absorbs red and far red light is phytochrome. So phytochrome, which is our option C, it absorbs which light? Red light. And it is involved in flaring and many other light mediated processes. So it absorbs red and far red light in plants for photosynthesis. Moving on to the next question, which one is a long day plant? So we just studied about short day plant. Now we'll study about long day plant. So long day plant actually need more of light period and less of dark period. Okay. So what is an example of long day plants? Long day plants example are spinach and sugar beet. When we studied the example for short day plants. Short day plants are glycine, max, and tobacco. Okay. So option D we have spinach. Tobacco, they have given option A, it is a long day, it is a short day plant. Option B is also a short day plant. Option D is a long day plant. Moving on to the next one, we have question number 16th, that the proteinaceous pigment which control activities controlled or concerned with light. We have a pigment which is made up of protein, and what is its function? It controls activities with respect to light. So, do you know of any such pigment? The answer for this particular question is phytochrome. We just studied that phytochrome, it absorbs light. So phytochrome is a colored protein pigment which is located where? It is located in plasma membrane. It is located in plasma membrane and it perceives the photoperiodic stimulus in leaves. Okay, so it, what is its function? It perceives the photoperiod stimulus. Okay, and where is the photoperiod stimulus activated or received? It, we studied it in the previous question only, that it is perceived in leaves. Okay, so it is present in the leaves and it actually perceives the photoperiod stimulus. Okay, and it exists in two interconvertible form, which is PR and PFR, which absorbs red and far red light. Okay, so moving on to the next one, is the method that renders the seed coat permeable to water so that embryo expansion is not physically retarded. So I taught you the concept that the seeds, they are in dormant phase. So we need to do some process or some activity so that they come out of dormant phase. So vernalization, we know that vernalization was the exposure of the seed to low temperature. Stratification was its exposure to low temperature as well as moisture, and the process in which we use physical force. Where is physical force used? Physical force is used to damage the seed coat. And this process right here is known as scarification. In scarification, we break the seed coat barrier, which can be done either by mechanical or chemical methods. So the response of different organism to environmental rhythm of light and darkness. Again, we have already studied this. So question number 18th, we have response to lightness and darkness, response to light period and dark period. This particular process is known as photoperiodism. Okay. What is vernalization? It is the exposure of the seed or seed coat to low temperature. Okay. So phototropism is the movement towards light. So photoperiodism is the effect of duration of light and darkness on the growth, development, and behavior of an organism. What will be the effect on phytochrome in a plant subjected to continuous red light? So we know what is phytochrome. Question number 19th. Phytochrome is a proteinaceous pigment and it absorbs red and infrared light. Okay, so if we expose phytochrome to too much of red light, too much of red light can cause PR, which absorbs red light, to PFR, and it is rapidly destroyed. So PR, which is phytochrome, is actually the amount decreases. Okay, so the level of phytochrome in this case decreases. So we need to make sure that we are not even exposing it to so much of light that we are damaging the phytochrome in general. Okay. Moving on to the next one. In short day plants, flowering is induced by, so we know plants can be either long day or short. Long day means that they need more of exposure to light to remain functional, whereas short day plants mean that they require more exposure to the dark period. So flowering can be introduced by what? So in a short day plant, they have given the definition. Short day plant is one that flowers on photoperiods that are shorter than the critical day length. Okay, so we have to have question number 20, photoperiod, which is shorter than the critical day length. Okay, and we studied the example for short day plant. Example for this is tobacco and glycine max. Okay. So next question. Flaring depend on cold temperature is known as vernalization. We studied this topic. Vernalization is the process in which this is a seed and it is in what dormant stage. So we expose it to cold temperature and then it can germinate. This particular seed can germinate, and after germination, it will also show flowering. So, the flowering that is dependent on cold temperature is actually vernalization. Vernalization is the method of inducing early flowering in plants by pre treatment of their seeds at what? Cold temperature or low temperature. So tendrils, this was our very first question. That tendrils, they exhibit twinning of tendrils. What is twinning of tendrils? That whenever a touch stimulus is applied to the tendrils show their movement towards that stimulus that is being applied, and this particular process is known as thigmotropism. So, twinning of tendrils is due to thigmotropism. That is the movement which is due to the contact with a foreign body. Thigmotropism, it is what is the hormone that mediates thigmotropism? It is auxin. So, auxin causes thigmotropism. Okay. So, which one increases in the absence of light? Question number 23. There is absence of light, and in the absence of light, the stem becomes excessively long due to etiolation. Okay, so in the absence of light, there is a process which is known as etiolation, due to which there is a increase in length, increase in length of stem, due to which internodes become larger. Okay. Which of the following hormone can replace vernalization? So, vernalization is what it is, the low temperature treatment for flowering in plants. So plants are treated with a very low temperature to promote germination. So scientists demonstrated that the application of gibberellins can replace the cold treatment or vernalization in many plants. So question number 24, vernalization can be replaced by which hormone, which is gibberellins. Okay. Moving on to the next one, phytochrome. What is phytochrome? It is a proteinaceous pigment, proteinaceous compound, which actually absorbs red and infrared light. So it is involved in photoperiodism. So question number 25, we have leaves which actually received stimulus for photoperiodism. So in the plasma membrane of these leaves, we have phytochrome, which absorbs red and infrared light, absorbs red and infrared light. Okay. So by stimulating cell division, they can. So phytochrome is involved in what? Phytochrome is involved in phototropism. Okay. Moving on to the next one, gibberellins. So what is the function of gibberellins? So this was asked twice. So question number 26th, gibberellins, they actually promote seed germination. We just discussed that vernalization in plants, it can be replaced by gibberellins, which help in the germination of seeds. So gibberellins, they actually promote seed germination. Okay. So there are some light sensitive seeds such as Listisca sativa, which is lettuce, which can germinate in absolute darkness. No light is needed. And if we add gibberellins from our side, they can germinate well. Okay. So moving on to the next question, we have abscisic acid. And what is the function of abscisic acid? We, in the beginning of the session, I explain to you the classification of plant growth regulators. So there was one group which actually promoted such activities, and there was the other one which actually decreased the cellular activity or cellular growth. So abscisic acid was under the category 2. So abscisic acid, it actually controls our leaf fall and dormancy, the activities which are opposite to that of cell growth. So abscisic acid, as the name suggests, causes abscission, due to which leaves falls. It also acts as a growth inhibitor, and it induces bud dormancy in various plants. So we have abscisic acid. It regulates what? It regulates abscission and dormancy. Okay. So, what are the functions of abscisic acid? It stimulates the closure of stomata, increases the tolerance of plants to various kinds of stress. So, it is known as stress hormones. Okay. So, moving on to the next one, this is also an important point that abscisic acid, it acts as an antagonist to gibberellic acid. So, we knew that gibberellic acid, it was involved in seed germination. Okay. Moving on to the next one, which of the following is not an inhibitory substance that governs seed dormancy? So, abscisic acid, I just told you, it inhibits seed germination. Phenolic acid also inhibits seed germination. Para-ascorbic acid also inhibits seed germination, but gibberellic acid, it actually promotes the seed germination. Okay. So it is not actually governing seed dormancy. So question number 28, we have gibberellic acid, which promotes seed germination. Okay, moving on to so they have told it right here that gibberellic acid, it actually breaks seed dormancy. It activates the synthesis of what? Alpha amylase. So gibberellic acid increases the function of alpha amylase, which breaks down starch into sugar. And the phytohormones, gibberellic acid has essential signaling functions in multiple processes during plant development. So, in rice, gibberellic acid signaling plays an important role in pikelet fertility. Okay. Name the plant growth regulator, which upon spraying on sugarcane crop increases the length of stem, thus increasing the yield of sugarcane crop. So, we have question number 29. This right here is a sugarcane crop, and on increasing on or on spraying this hormone, the height increases. So this is the function of gibberellins. Okay, so gibberellins are another kind of promoter. They promote cell growth and they cause an increase in length axis, and it is used to increase the length of grape stalks. And it also causes fruits like apple to elongate and improve its shape. Okay, so they are also used to speed up malting process in brewery industry. So sugarcane, they store carbohydrate as sugar in their stems. So spraying sugarcane crop with gibberellins increases the length of stem, thus increasing the yield by as much as 20 tons per acre. Okay. So next question we have that the removal of shoot tips is very useful technique to produce the production of tea leaves. How is this actually possible? So we have auxins. Okay. So auxins is actually indole 3 acetic acid. So they have growth regulating property. They are produced by the growing apices of stems and roots. Okay. And from there, they migrate to the regions of their action. All these, they help in initiate rooting in stem cutting. And in most of the higher plant, the growing apical bud, it inhibits the growth of lateral buds. Why is this so? Because the bud, it produces auxin. And this phenomena right here is known as apical dominance. So removal of shoot tips, which is known as decapitation, it results in the growth of lateral buds. And this is widely used in tea plantation. So right here we have our apical bud. This is apical bud. This apical bud actually produces auxin, and this auxin helps in this vertical growth. And this vertical growth, it inhibits the lateral growth. So if we cut the apical tips, if we cut the apical tips, there will be the auxin production will decrease, and hence the lateral growth for these particular plants will increase. Okay, moving on to the next one. We have, in order to increase the yield of sugarcane crops, which of the following plant growth regulator should be sprayed? We just read this topic right here. So spraying the sugarcane crop with gibberellins, it can actually increase the length of stem, thus increasing the yield by as much as how much? 20 tons per acre. Okay. So gibberellins, they regulate various developmental processes, including stem elongation, germination, dormancy, flowering, flower development, and leaf and fruit senescence. When gibberellin is sprayed on sugarcane crop, the length of stem increases. And this was discovered by Kurosawa, who was a Japanese botanist. Okay, so there are various types of gibberellins that are being isolated, and they have been named as GA1, GA2, GA3, and so on. So GA3 is the gibberellic acid, which is most commonly used and also studied. Okay, so it takes very long time for pineapple plants to produce flower, which combination of hormones can be applied to artificially induce flowering in pineapple plant throughout the year to increase yield. So this right here is which question? Question number 32. So we have a pineapple plant, and what do we want to do? We want to produce flowers. So how do we hasten this process? In order to hasten this process, the pineapple plant flowers only one, and one plant will only produce one pineapple. Then it dies, but before it dies, it also produces offspring. So, auxine and ethylene can be applied to artificially induce flowering in plants, in pineapple plant to increase yield throughout the year. Applying auxine, it mimics the action of ethylene by stimulating ethylene formation, and that ethylene, not auxine. Actually, ethylene causes the pineapple to flower because ethylene it initiates flowering and it synchronizes fruit set in plant. So, what is the hormone that is responsible for flowering? It is actually ethylene. It produces flower. But in order to increase the amount of ethylene, we are giving auxine because auxine it stimulates and it increases ethylene formation. Okay, moving on to the next one. Fruit and leaf drop at early stages can be prevented by the application of. So, question number 33 we have that we have to prevent fruit and leaf drop. So, what hormone are we going to use? So, we studied about auxin here. So, auxins, they let us study a bit about all the hormones. So, they have told us that auxins, they help to initiate rooting and stem cutting. And auxins, they promote flowering in which plants? They promote flowering in pineapple, which was just the previous question. And they also help to prevent fruit and leaf drop at early stages, but they promote the abscission of older, mature leaves and plants. So, auxin, it would prevent fruit and leaf drop for which fruits and leaves which have not matured yet. So, with this, we can give them more time. So, there is increased time for it to mature. But in case of the fruits and leaves that have been already matured, so for matured leaves and fruits, it helps in their abscission. Okay, so it is a relative concept. So auxins they help to prevent premature leaf and fruit fall. So the avena culture, we have next question, which is question number 34. So do you know what is avena culture? So avena culture, it is a culture through which auxin has been the bioassay of indole IAA. So, what is IAA? It is indole 3 acetic acid. So, with the help of Femina culture, we can use it as a bioassay for auxins. And what is an example of auxin? It is IAA, which is indole acetic acid. Okay, that is given right here. That auxins we can have IAA and IBA. So IAA and IBA, these are natural auxins, indole acetic acid and indole butyric acid. They have been isolated from plants. Whereas there are also synthetic auxins. And what are the examples of synthetic auxin? It is NAA, which is naphthalene acetic acid, and 2,4-D, which is 2,4-dichlorophenoxy acetic acid. So they are examples of synthetic auxin. So auxins can either be natural or they can be synthetic. Natural, we have IAA, which is indole acetic acid, or IBA, which is indole butyric acid. Synthetic, we have NAA, which is naphthenic acid, and we have 2,4-D, which is our 2,4-dichlorophenoxy acetic acid. Ravenna culture is how can we, in which plants we demonstrate Avena culture? So auxin has been clearly demonstrated in the leaf sheath or folioptile of an oat plant. So oat plant, we have the scientific name as Avena sativa. Okay. So Avena sativa is our scientific name for oat plant. And with this, we have the name given as Avena culture. So this plant coleoptile, it has been used for testing the hormone auxin, which is participating in the growth of plant. So we have our next question, which is question number 35. This is the last one. So what causes a green plant exposed to a light on only one side to bend towards the source of light? So this question number 35, we have this plant and it is being exposed to sunlight only on a single side. So this plant is exposed to sunlight on a single side. And this stem, it bends towards source of light. So this is what? This is due to auxin. Okay. So we have auxin. What is auxin? Auxin actually helps in the cell elongation. Okay. So it accumulates on shaded side, which results in more elongation of cells towards shaded side of plant, and this causes the growth of light towards the source of light. Okay. So we have this right we have. So it was observed by whom? It was observed by Charles Darwin and his son Francis Darwin that when they observed that the coleoptiles of canary grass they responded to unilateral illumination by growing towards the light source. Okay, and this right here is due to the function of auxin. So with this, we are done with our questions for the chapter plant growth and development. Thank you.